Serena Council have all shown that there are persistent disturbances that are still seen in adolescence and correlate with impaired total IQ and cognition. So these are very enduring disturbances in development. So what might be the origin of these developmental disturbances? The overriding hypothesis among investigators is that this is related to hypoxemia. Kelly, are we still good here? Can you hear?
Yep, we're good.
All right, just checking in because I'm talking to a screen and have no idea if there's anyone out there.
You have plenty, yeah, you have quite a few.
All right, so fetal hypoxemia and CHD, when you think about fetal brain oxygen consumption, it's really quite a paradox. Now, why would the brain be hypoxemic in kids with congenital heart disease? We know that the fetal brain is essentially hypoxemic to begin with. Joe Volpe, my mentor in Boston, showed back in the 1980s in an elegant paper in Annals of Neurology that preterm infants had cerebral blood flow, which was only about 20% of that of adults. And various estimates of from animal studies have estimated that fetal and even adult mammalian brain, that the deep white matter structures are very low in oxygen tension, and particularly in the fetus are about 1 to 5%, so much lower than room air at 20%. Despite the fact that the fetal brain is so hypoxemic at baseline, we know that the fetal brain is a voracious consumer of oxygen. It utilizes about 25% of total ventricular outflows. And it consumes roughly 50% of the total systemic oxygen. So the brain is just very aggressively consuming oxygen relative to other tissues. And this is particularly true in the third trimester when the brain is actively beginning to establish dendritic arbors in synaptogenesis and myelination. So we see in CHD that there are, as we will look in further detail, significant reductions in oxygen saturations, and it's estimated that the fetal brain consumption is reduced by about 1/3 in CHD. So let's look at that data in more detail now. I apologize for the complexity of this slide. We could probably spend the entire hour just thinking about this slide. But just briefly, I want you to remember that the placental oxygen traveling via the umbilical vein, via the ductus venosus, reaches the right atrium, crosses to the left atrium, and then exits via the ascending aorta to the brain. And at left, you can see this nice summary of an article, summary diagram from Mike Seed's lab in Toronto, where you can see that brain oxygenation is, you can see saturation scale here on the left, and the brain is well oxygenated. By contrast, in transposition, hypoplastic left heart, tetralogy, and flow, there are various significant reductions in oxygen delivery due to various mixing defects that impair the oxygenation. And in this paper, Mike Seed began to look at cerebral oxygen consumption and how it's related to brain growth. It's a very elegant paper. If you haven't read it, I encourage you to take a look at it. And so you can see on the left graph that cerebral oxygen consumption is reduced significantly in children, fetuses with congenital heart disease. These are near-term, late gestation fetal controls compared to the kids with congenital heart disease. Again, these are all fetal measurements now. And there's a reduction in brain volume, which is significant in the CHD group compared to the controls. And in the lower left-hand graph, you can see that there's an inverse relationship between cerebral oxygen consumption and brain weights. So as oxygen consumption falls in congenital heart disease, you see this decline in brain weight. So it's a very robust and highly significant relationship. In this paper, they defined a number of really interesting factors that contribute to this reduction in fetal cerebral oxygen consumption. It should be noted first that the placental oxygen delivery is reduced. So if you look at umbilical vein oxygen content, it's down. And this is not surprising because we're now recognizing that placental maturation in kids with CHD is not normal. The developmental programs that regulate maturation of the vasculature in the placenta also regulate the congenital heart. And so, not surprisingly, placental delivery of oxygen is already impaired. And that translates to about a 10% reduction in the oxygenation that's measured in the ascending aorta, which is supplying blood to the brain. Now, quite interestingly, and this has been a real puzzle for the field, is the question, is there cerebral ischemia affecting reduced brain blood flow, or is it reduced oxygen content? And from this study, they've observed that there were no decreases in cerebral blood flow. So that's a very important concept, I think. And the major finding, though, is that there was decreased cerebral oxygen delivery, and particularly decreased venous oxygen consumption, which is highly significant and reduced by about 30%. And so this is telling us that cerebral oxygen content, oxygen consumption is down, and it's not related to cerebral ischemia. More recently, again, another study from Mette Lauritsen in Denmark, a very elegant study in which they attempted to make fetal MRI measurements of total brain oxygenation. So using MRI T2 star, which is a measure of tissue deoxyhemoglobin concentration, they did serial scans of the fetus at 32 and 37 weeks. They had 28 controls, 15 children with major congenital heart disease. And what you can see in the graph is that they made two measurements. In blue, the blue solid line shows you the mean cerebral oxygenation and how that changes over time in fetal life. And you can see that as we approach near term, around 38 weeks, oxygen content in the brain is gradually declining compared to earlier in development. And it's much more pronounced, as you can see in the red lines and the red dots, in those children with congenital heart disease. Now, if we look at newborns, this is studies from Serena Council and David Edwards in London, where they have a scanner right in the NICU. Serena's group looked at cerebral oxygen delivery and how that correlated with disturbances in brain growth. Now, in the upper panel, you can see that there are diffuse, highly significant reductions in the blue areas on the brain scans are showing you areas that are reduced in brain volume for the children with congenital heart disease relative to controlled term neonates. And when they looked at indices of total brain volume, cortical, white matter, cerebellar, and deep gray volumes in the table, you can see that they were all significantly correlated with reduced cerebral oxygen delivery. So again, confirmation that there's a persistent disturbances in brain growth that are present in the term neonates and correlate with decreased oxygen delivery. This is a very elegant study that I wanted to share, which takes us back to this concept of that I discussed earlier about the reduced complexity of the arbor of individual neurons, this dismaturation of neurons. And so taking off from that study we published, Serena sought to use the so-called Orientation Dispersion Index, or the ODI, as a measure of complexity of neuronal processes. You know, what's the total density of neuronal processes estimated by the ODI, and how does that compare with cerebral oxygen delivery? And you can see at left that the areas in red are showing the areas where there's a significant deviation from controls in terms of the overall density of neuronal processes. And at the graph is showing you that the complexity of the dendritic arbor, the density of neuronal processes within gray matter, cortical gray matter, falls off as oxygen delivery to the neonate is lower. So there's a very strong correlation between maturation of cerebral cortical neuronal complexity and oxygen delivery to the brain. So with that introduction, then I wanted to share with you some of our own data, which tries to get at this question of how hypoxemia affects fetal brain development. The primary model that we use in my lab is the preterm fetal sheep model. Here you see a little flock of sheep that, if you ever have the pleasure of being in the Oregon wine country, as I was on this day, there was a nice, at one of the very nice hotels, I stumbled into this little collection of sheep and wanted to take them home with me, but this whole, they wanted $7,000 for these sheep. So my wife said no, and all I have is this picture to share with you. But I'd like to share with you that the cerebral dismaturation story has been driven by two key postdocs in the lab over the years. Justin Dean, who's now on the faculty in Auckland with Alistair Gunn in New Zealand, and Evelyn McClendon, who are still in the lab. And I want to just focus on one of the more recent studies we published to try to begin to understand how hypoxemia might disrupt learning and memory in both preterm infants and children with congenital heart disease. I became interested in this problem because of the clinical observations that volumetric studies of the hippocampus found that preterm survivors had reduced hippocampal growth. A lot of that data coming from Terry Inder's group. And that reductions in hippocampal volumes were associated with long-term abnormal working memory, problems with attention and socialization, which Terry's group studied in a number of elegant papers highlighted here. And these reductions in hippocampal volumes in preterm survivors by adolescents correlate with significant cognitive delay. So we have been looking over the years at how hypoxemia, transient hypoxemia, a brief insult, can disrupt maturation of multiple structures that are involved with learning and memory, including the cortex, the caudate, and as I'll show you in this study, the hippocampus. The model that we use is one in which we operate an instrument, a fetal sheep, at 2/3 of gestation. So they're roughly equivalent to a 26 to 28-week infant. They survive for 3 days, and then they're made either hypoxic or hypoxic ischemic. And then they survive for 4 weeks, after which we analyze the brains both by various anatomical methods and physiology, which I'll share with you shortly. So this little cartoon here shows you that the hypoxic fetus is a
Dr. Back, we can see that. Dr. Back, we don't see that
yes?
slide. We don't see that slide.
Okay, let me try again. Can you see it now?
No, we still see your picture with the sheep.
Really?
Yeah.
How about now?
No, still.
The slide suddenly stopped moving. Oh dear. Well.
Can you try maybe double clicking on the next slide?
Yeah, that's what I've been doing.
Yeah. It might just be frozen.
Let's see. Previous slides. I don't know. I mean, nothing happened on this end to indicate that we had a problem with the slides, but clearly, so I'm moving them, but you're not seeing anything. You're still stuck on the picture of the sheep.
Right.
Oh dear. What do you suggest? Should I exit out and try to reload?
I would.
All right. I'm sorry about that.
No problem.
So now let's see. Let's see. Open the share tray. I'm going to have to, I guess, open the talk again and see if it seems to take some time before the talk ends up in the share tray. Like, there's no way to drag it in, I guess. Is that right? It just has to appear on its own. All right, I'm going to close the talk again and open PowerPoint, open the talk. Let's see. Yeah, I've never had problems with Zoom or other platforms, but this one is really, I need to have a talk with Bill Gates about this. It's just not showing up in the share tray. Do you have any suggestions, Kelly, or?
Make sure your camera is off.
Make sure my camera is off.
Yeah.
Kelly, do you have a copy of the slides?
I do not. If you want to send me a copy, maybe I can open them up.
That would be a good idea.
Well, that was the problem, is that they were so large they wouldn't. I tried emailing them to you last night. Oh, for goodness sakes. Open the share tray.
I know you're working on an Apple.
Which shouldn't matter. Oh, goodness. This is really taking a while because of all these technical problems. Okay. It's supposed to just appear in the share tray, right? I've opened the talk. It's on my desktop.
Did you double click it?
Yeah, I've double clicked it. I can see it. Does it have to be? I mean, if I put it into presenter mode, then I won't be able to see the. Maybe you should take me off of share and put me on again, Kelly. Maybe
Okay.
that's it. I don't.
Where did you go? Dr. Carnati, it looks like he went completely off.
Okay.
Hello, Dr. Bass. Can you log all the way back in again? Maybe we can fix it that way.
Yeah, well, that's what I'm trying to do now. For some reason, you kicked me out of the session.
So we can see you now.
Okay. So I have to share. I need to be able to share my slides now. So can you? All right. Now there's the talk.
Share.
Let's see. Hold on. I just need to get to the there. Okay. All right, so I've double clicked. I can see the slides. Can you see them?
And you have to stop your camera, actually, I think.
Oh. All right. Let me see. Okay. I've
Yeah,
stopped the camera. Now I will put it back on presentation mode. Can you see it now?
No, not yet.
Oh, God. Bill Gates is really good to hear from me.
That's okay. We can.
Is there anything else I should be doing?
It's weird. I can see the slides on my phone, but I don't think on a computer.
So I have my camera off, and
Double click on your first slide. Yeah, now we can see.
now you can see.
Yeah, the twin pregnancy model.
Yep.
That.
Okay. I'm going to hang up the phone. All right. So this was the twin pregnancy model I was mentioning, where you can see that the hypoxic fetus is made hypoxic for 30 minutes. The HI fetus is both hypoxic and ischemic. And you can see that arterial content rapidly falls in the fetus and actually stays much lower in the hypoxic fetus relative to the hypoxic ischemic fetuses, which I'll come to in a moment. You can see that we generate reduced hippocampal growth, as we see in infants, preterm survivors. The hypoxic group in the blue show significantly reduced hippocampal growth. The interesting thing about the hippocampal neurons is that they actually show very complex changes in their neuronal arborization. You can see in the lower panel the actual Golgi staining, where the hypoxic neurons are very hypercomplex looking, particularly in the basal dendritic arbors. Not showing here are the apical dendrites, which are actually less complex. So they're very robust changes in hippocampal neuronal anatomy that happen in response to the hypoxemia. And you can see that this is oxygen dependent. And if you look at the complexity of the neurons as a function of low or high oxygen, on the left, you can see high oxygen. We can measure the oxygen content of each fetus throughout the experiment. And you can see that the high oxygen fetuses really don't differ from controls, but it's the fetuses that show lower oxygen content from their ABGs during the experiment that show the disturbances in neuronal complexity. What I think is particularly sobering is that we can actually, when we plot the drop in oxygen content as a function of the complexity of neurons, you can see that there's a very striking association, highly significant, which basically tells us that depending upon how hypoxic a fetus becomes, you can determine how disrupted brain maturation is going to be. And I remind you, this is only 30 minutes of moderate hypoxia. And so I think this has some very sobering implications when we start thinking about our babies in the NICU and the kids with congenital heart disease, because presumably the more severe the mixing defect and the more hypoxic those infants are in utero, the more the disturbances in neuronal maturation will occur. I'll skip this in the interest of time, but basically we do circuit recordings where the brains are rapidly collected when the fetal brains are collected, they're rapidly immersed in cold artificial CSF and rushed to the lab so that we can actually record from individual brain cells to see what the fetal maturation looks like physiologically. And in this slide, you can see that hypoxia significantly reduces action potential firing in hippocampal CA1 pyramidal neurons. And I'll focus on the graph at the very far right, where you can see the hypoxic neurons in blue are significantly reduced in terms of their number of action potentials they fire compared to the controls. Now, if we look at a cellular measure of learning and memory, so-called long-term potentiation, in which if you stimulate with a rapid train of impulses a neuron in the CA1 hippocampus, you can generate a phenomena called long-term potentiation, where there is an elevation in the field potentials of these cells. And that's illustrated in panel C, where you can see that LTP is markedly reduced in blue for the hypoxic animals relative to the controls, or even the HI animals. So hypoxic animals, which sustain the lowest falls in oxygenation, show these significant reductions in a cellular correlate of learning and memory, LTP. In the interest of time, I won't go into the paired pulse ratio in great detail, only to say that this is a measure of presynaptic glutamate release. And the higher this value is, the more impaired glutamate release would be. And you can see again in the hypoxic animals, very significant elevations in this PPR ratio, which is an indication of impaired presynaptic glutamate release. So just to summarize this, a transient insult, 25 minutes, 30 minutes of hypoxia is sufficient to cause enduring changes in maturation of neurons in the fetal hippocampus, both as defined by their anatomy and their physiology. We see presynaptically, there's decreased glutamate release. Postsynaptically, decreased action potential firing and changes in long-term potentiation. And I think what this is telling us is that the fetal brain is exquisitely sensitive to hypoxia. And this causes profound disturbances in neuronal maturation, which occur in regions that are integral for long-term cognitive development and learning and memory. And what we don't really know at this point is how these disturbances may occur in the kids with congenital heart disease. The fetal brain, we don't have animal models at this time that can generate chronic hypoxia that looks like what we see in congenital heart disease. And I think the studies I've just shown you are tantalizing in that they suggest that even a transient hypoxic insult is enough to disrupt the maturation of the brain. And we hope that in years to come, we're going to be able to model this more elegantly, to understand the mechanisms behind this, which will hopefully lead to improved therapies. But I want to close now with just a few slides to bring this back to the kind of clinical management and think about what can we do at this time that might make a difference for kids with congenital heart disease. So the first notion is just simply that prenatal diagnosis, diagnosing congenital heart disease in utero, really allows cardiologists, surgeons, neonatologists to plan for the delivery and to stabilize these babies. And if you look at the distribution of brain injury that was plotted in these graphs.
Dr. Bach, I think we lag behind by one slide, I think 1 or 2 slides. I think right now we are seeing the hypoxemia and cerebral dysmaturation slide.
Let me try double
the next one, maybe the next one. I think you're talking about the next one.
Okay, can you see now the brain injury, what makes a difference?
Not yet. We still see that same. Yeah, now we can see, yes. Thank you.
Now we can see it. All right. I'll just keep double clicking and hope that works. So the point of this slide is that prenatal diagnosis makes an enormous difference in terms of the burden of brain injury that these patients sustain. And so if you look at B, the majority of these patients did very well in terms of the burden of brain injury compared with A, where congenital heart disease was postnatally diagnosed by a preoperative MRI. And there you can see that there are children with significantly more brain injury. So if you look at the lower panels, you can see that the orange line indicates the response to a prenatal diagnosis versus the black line, which is postnatal diagnosis. And at left, you're just looking at white matter maturation as measured by fractional anisotropy, which we expect to increase over time as the white matter matures. And you can see that it's increasing better for the kids who are prenatally diagnosed with their heart disease compared to the postnatal. And similarly, if you look at ADC, which is a measure of white matter or gray matter injury in this case, how watery the brain is, there's lower ADC, which is correlated with improved brain maturation in the kids with prenatal diagnosis. So again, just the timing of diagnosis makes a difference. Can you see the next slide?
Not yet.
Not yet. Let me see.
Yeah, we can see now.
Okay. So these are some interesting studies that have been done by Kathy Limperopoulos' group at DC Children's Hospital, looking at the impact of maternal stress on fetal brain maturation. This is becoming a hot topic, I think, in neonatology and in congenital heart disease management. We know that psychological stress is very common after a diagnosis of congenital heart disease, and that pregnant women suffer depression, anxiety, and a good deal of stress. And this is associated with cortical thinning, reductions in growth of the hippocampus and amygdala. And so what you're looking at here are views of the hippocampus at left and right. And the blue areas are showing you the areas where there's a significant reduction in hippocampal growth, and on the right, cerebellar growth. So, and this is all highly correlated with the degree of maternal stress. So this is quite a, can you see the next slide, maternal hyperoxygenation?
No.
No. Oh dear. Let me see. If I double click, I move ahead 2 slides. I don't know why we're having a problem again. You can't see the maternal hyperoxygenation slide?
No, not yet.
Oh dear.
Yeah, now we can see. Yeah.
Okay. It seems to have quite a lag for some reason. So here what you're looking at, yeah, you're on Eastern time. I'm in Oregon. So maternal hyperoxygenation is being considered as a potential therapy now for kids with congenital heart disease. And how this is all going to play out is it's not really clear. Whether we would chronically make the mothers hyperoxic during pregnancy to deliver more oxygen to the fetus to try to reverse the effects of hypoxemia. These are things that are being considered. And you can see in the green box that their measurements were being made of umbilical venous oxygen saturation using T2 measurements. And after hyperoxygenation, the fetal oxygenation in the kids with congenital heart disease, the fetuses, were significantly increased, although this had no effect on normal controls, which is interesting. So the next slide, which hopefully you will eventually see, looks at maternal hyperoxygenation in a little bit more detail. These are studies from CHOP, where they looked at cerebrovascular resistance. And multiple groups have shown that the CBR, the cerebrovascular resistance, is reduced in kids with congenital heart disease, apparently as an attempt to increase oxygen delivery to meet the greater demands for brain growth in the 3rd trimester. So in this study, can you see the slide yet?
Yeah, we can see.
Okay, maternal, by doing maternal hyperoxygenation, you can see it on the left graph, just what cerebral vascular resistance looks like in the CHD group relative to these, again, Z-scores, so they're compared to a normal control population. You can see how significantly the vascular resistance drops off between 25 and 40 weeks. And then at right, you can see that with hyperoxygenation, the fetus actually does respond, and that drop off in oxygenation is blunted. And so there's an improved response. So this all suggests that the fetus has physiological responses to hypoxemia, which are abnormal, that hyperoxygenation does at least show proof of concept that hypoxemia is an important factor that's disrupting cerebral physiology, and it remains to be seen whether that might have an impact on long-term brain development. I think there's still a lot that needs to be worked out before we think about this as a therapy. So I want to close with just a couple of thoughts about socioeconomic status, and just what's the impact of maternal education on brain injury. This is an interesting study recently published by Steve Miller's group, where they asked the question, if you look at preterm infants and their expected IQ, their measured IQ at 4.5 years, what's the impact of maternal education on that? So if you look at the orange solid bar, that is the IQ of babies that didn't have brain injury and whose mothers had a postgraduate degree. And you can see in the blue bars that if your mother had an undergraduate degree but not a postgraduate degree, your IQ trended significantly lower, as did the solid black line for the children where the parents had primary or secondary school education. So there's already an impact, as we know, of education on the maternal education on IQ. Now, the dotted lines show what's happening in response to brain injury. And what you can see, if you look again at the orange lines, those mothers who gave birth to babies who sustained brain injury, their IQ difference compared to controls was essentially blunted or mitigated. So higher socioeconomic status education blunted that difference, but that was not seen. If you look at the difference between the dashed black line for the parents with primary or secondary education compared to the solid black line, there's a substantial difference there in IQ. So environment seems to matter, and whether this is all genetically driven or driven by other factors, including the environment in which the child is raised, is an area that's, I think, a very hot topic right now. And I'm going to close with this slide. We have very little information now about congenital heart disease and what really makes the difference in terms of the environment. This was a small study that I found from the London group, where they looked at cognitive function in toddlers with congenital heart disease, and they employed a cognitively stimulating parenting scale as a measure of how stimulating the home environment was, and showed that there was, if you had a low stimulating environment, as shown by the blue dots, that the mean cognitive scores for those babies was about 20 points lower than the infants with higher parenting scales. So we've suspected for a long time that environmental enrichment is probably important. And for kids with congenital heart disease, clearly more needs to be done, both looking at socioeconomic status as well as the home environment. But I would suspect that that's probably going to be an important and low-hanging fruit that we could implement as a way to improve cognitive outcomes in these children. Now, I'm going to close just simply with this slide, thinking about some of the low-hanging fruit that we might be able to consider now as clinicians to reduce cerebral dysmaturation, improve cognitive outcomes in children with congenital heart disease. Clearly, just having a prenatal diagnosis can make a difference in terms of reducing the burden of brain injury. addressing maternal stress during pregnancy looks like a critical and important factor. Aggressively treating low oxygen states is going to be a challenge prenatally, but potentially there might be an impact of this postnatally. And then environmental enrichment and things like when to begin physical and occupational therapy. Should they be started in the NICU? Should all the babies be lifting weights when they're this would be something Dr. Carnardi can start his own gym in the Cleveland Clinic for these little ones. So there's a lot of tantalizing future directions that I think we should be embracing currently. And so with that, I'm going to close. I thank you for all your patience for all the disruptions during this talk, all the technical glitches. And I'm glad we made it to the end. And these are the people in the lab, if you can see them, that make the difference, so that I can get up here and give the talks, my collaborators and my support. And so I'll close there, and I thank you for your attention.
Thank you, Dr. Bach. Thank you so much. That's a wonderful talk. Yeah, again, despite tech glitches, this is definitely excellent talk. And great work. Congratulations. I mean, that's a lot of yeah. All right, there is a question here. How much of the brain effects for kids, what do you think, is it due to hypoxia versus the other factors you mentioned, the genetic, environmental, and developmental factors? How much is the contribution of these factors? Is it mainly the hypoxia, or is it?
Well, I think the message really is that the insults to the brain begin in utero, and I think we did not really appreciate just how profound that impact is. Undoubtedly, environmental factors and genetic factors are all going to play a role in terms of the interaction with hypoxemia to either exacerbate or lessen the effect. And that's why I wanted to mention things like maternal stress during pregnancy. We certainly know from recent studies from Steve Miller and Terry Inder that in preterm infants in the NICU, just the burden of painful procedures has a profound effect on cognitive outcomes in these babies. And those are really very robust findings that tell us that stress, the environment in the NICU are all very important factors that influence the burden of brain injury. So does that address that question?
Yeah, I think yes. Thank you.
Okay, great.
Role of nutrition. Are there? I know that oxygenation. That is very fascinating. I think I'm looking forward to that. Any nutritional mitigation we can do? Any specific nutrient or anything we can? Is it studied in this situation?
Yeah, we're just starting to think about this with congenital heart disease. As you know, this has been a burning issue and a key issue for the preterm infants. And again, studies from Steve Miller's group, for example, have shown how important nutrition is on mitigating white matter injury, for example. The babies with better nutrition do significantly better. And so, of course, the kids with congenital heart disease probably, it's highly likely that there is disturbances both in oxygenation and cerebral metabolism, glucose metabolism. These babies these fetuses are hypoxemic. In utero, they're largely using anaerobic metabolism to generate glucose, to metabolize glucose to lactate rather than through the Krebs cycle. and that could be all impaired by the hypoxemia. So I think whether we can have an impact on nutrition in utero is still a burning issue and a really important question. And certainly postnatally, we should be thinking about this with these kids, because I think that postnatally improved nutrition, improved oxygenation could potentially blunt the long-term brain dysmaturation in these kids to some extent, and potentially reverse it. You know, I think we just don't know at this point how reversible this is. So
that's great.
Does that answer that?
Yes, yeah, thank you. Any role for any stem cells which is used in the other neurological injury? Any of those studies available?
Yeah. You know, the whole notion that we can repair the brain with stem cells, I think, is still a challenging area. Obviously, lots of people are studying this. My lab has had a long-standing interest in what are the barriers to regeneration and repair? What prevents the brain from repairing after injury? And we've been focusing particularly on the extracellular matrix as kind of this, what people used to think of as the glue that holds the cells together. And we've had papers now over a number of years, going back to a Nature Medicine paper in 2005. So we've been studying this for about 15 years. And hyaluronic acid, which is the backbone of the extracellular matrix, we've discovered is a very important molecule which blocks white matter maturation. We had a paper in JCI a couple of years ago showing that oligodendrocyte progenitor maturation is blocked by specific forms of hyaluronic acid. So there are responses to brain injury that keep cells in a more stem cell-like state. So we think that the role of hyaluronic acid, for example, is to keep cells, maintain cells as stem cells until they're ready to start differentiating during developmental programming. And so the question arises whether, if you put stem cells into a hostile environment where there's brain injury, will those cells differentiate, or will they just sit there because of inhibitory cues within the lesions? So I think there's still quite a lot to be done. Most of the studies in stem cells have looked at healthy brains that they've injected them into, and the jury is still out on that one. But that's a great question, one that I think about a lot.
Let me see if you have some. So because of this role for hyperoxemia, I mean hyperoxygenation. So yes. Similarly, like smoking, is there a push for something like we should be pushing for advising these mothers, especially with the cardiac kids?
Yeah, that's a great question. I haven't given that enough thought. One of my colleagues, Cindy McAvoy, is a neonatologist here at OHSU, and she's been doing extensive studies on the impact of maternal smoking on lung development. And there's no question that there's dysmaturation of the lungs as a result of maternal smoking. We haven't looked at the brain. Most of her studies are being done in non-human primates, so they're very close to human. I think one could well expect that maternal smoking would be very deleterious to the development of a fetus with congenital heart disease. That combination of fetal hypoxemia just from the heart disease, coupled with the hypoxemia from maternal smoking, and all the other vasoactive factors that might be affecting placental oxygen delivery, that could be a really deleterious combination. I think you should get a fellow to start studying that. That would be a great project to look at outcomes in kids with congenital heart disease as a function of maternal smoking. Yeah.
Yeah.
Excellent question.
Thank you. All right. I think I think that we didn't have any more questions. Thank you, Dr. Bach.
All
Thank
right.
you very much, and thanks for your time. We'd like to see you again.
My pleasure. Hopefully next time in person so we can get rid of Teams. But thank you all for your patience today, and I'm glad we were able to get through the talk.
No, it's great.
Have a really good weekend.
Yeah,
and
thank
enjoy
you. You
the
too.
beginning of spring.
Yeah.
Okay.
Thank you.
Bye-bye.
Thank you, everybody. Have a nice weekend. Thank you.